So okay, so we are going to discuss part B, chapter 3, homeostasis. We are going to look at question 6 until question 10. So for question 6a, it gives you figure 6 that represent a vertical section through a mammalian kidney. So can the first person in the list, Umar, to answer question number 1?
Number 1. Name the region M and the structure N. M is cortex, N is pelvis.
Structure. And if you look at the question here, N is actually, it is ureter. Kalau tengok dalam pada nota awak, pelvis atas lagi. Pelvis yang lagi besar, dia punya funnel dia tu lagi besar. Boleh lah kot. Kalau dekat sini ureter lah. If here, renal pelvis. Okay. Sebab dia tunjuk kat situ, maybe boleh lah juga kot dia, we accept renal pelvis. Okay, the bigger funnel is the renal pelvis. The more, narrower tubule is the ureter. Okay.
Okay, madam.
Okay. Alright. So the next question. Okay, so but before we move on to the next part, okay, so you know that the kidney, it is divided into regions. Okay, the cortex region is the outer region, and the region here is the medulla region. The medulla region is divided into two sections, which is the outer medulla and also the inner medulla. Okay, so here is the inner medulla, here is the outer medulla. Okay. Nampak kat situ. Okay, so here. The outer medulla. Nampak juga. Saya nampak. Okay. Tak apalah kat sini. Okay, so the outer medulla. Dia lagging, sorry Dia lagging. Outer medulla and also inner medulla. So the cortex region will have the glomerulus, will have the Bowman capsule, proximal tubule and distal tubule in the cortex region. In the medulla region, we'll have the loop of Henle and also the collecting ducts. Okay. So those are the component of the nephron in different region of the kidney. So number 2, Sharifah Nur Aina.
Number 2, explain how glomerular filtrate is formed. I answered hydrostatic blood pressure in the glomerular capillaries is higher than in the other capillaries. High pressure due to the high resistance to outflow presented by the efferent arteriole, which is smaller in diameter than the efferent arteriole. Then moderate permeability of glomerular capillaries due to numerous small pores between the endothelial cells make the glomerulus capillaries more porous than typical capillaries.
And causes the process of ultrafiltration to occur. You can also include that in your answer. So you have most of the point there, okay? So most of the point is in your answer. So this question is actually, if you look at the mark, it is 5 marks, okay? So you have to include a lot of explanation in your answer, okay? So but okay, so but at your level, okay, during exam or during test, there will be less tendency of the question that will be of 5 marks or 4 marks. Selalunya 1 markah, 2 markah, 3 markah, kadang-kadang 4. Macam tu, okay, 5 markah tak ada. So ini just sebagai latihan lah. So latihan so that you can add more information in your answer for your revision. Okay. So basically in the question, so jawapan Sharifah tadi dah okay dah you got most of the point. glomerular filtrate is the filtrate that is formed due to the process of ultrafiltration that occurs between the glomerulus and the Bowman capsule. So in the glomerulus, there will be high pressure due to what you mentioned tadi, the diameter of the afferent arteriole is wider or bigger compared to the efferent. Tadi awak sebut vice versa lah kan. Efferent has a smaller diameter compared to the afferent. Okay, can be both ways. Asalkan you mention the different in diameter. Afferent bigger diameter, efferent smaller diameter. So this create a high blood pressure in the glomerulus, causing fluid. Okay, causing fluid in the blood in the glomerulus to enter into the Bowman capsule, forming the filtrate. Okay, filtrate is formed once the fluid enters into the Bowman capsule. Okay, so and also due to the structure of the glomerulus and the Bowman capsule that is very thin. Okay, it is one cell thick, and then the glomerulus is thin, the Bowman capsule is also thin, and they also have pores. Okay, the cell have pores in between. Okay, they are porous, and then you also have the formation of filtration gaps. Okay, filtration gaps that allows substance such as urea, water, glucose, uric acid, the sodium ion, the chloride ion to enter into the filtrate from the blood in the glomerulus. Okay, so those are the answer. Okay, so saya akan ulang balik semula so that you can get the answer. I'm not going to write the saya share semula lah. Okay, so point number 1. Okay, point number 1, high blood pressure in the glomerulus. Point number 2, due to what? Due to the difference in diameter of afferent that is wider compared to the efferent arteriole. Point number 2. Point number 3 is that the fluid in the Bowman's capsule, okay, Bowman's capsule will be in lower pressure, okay, lower pressure compared to the blood pressure in the glomerulus. Okay, so next, this causes fluid from the glomerulus to enter into the Bowman capsule through the process of ultrafiltration. Okay, through the process of ultrafiltration. And then the next one is, if you mention, the endothelium of blood vessels or the endothelium of the glomerulus, okay, have gaps or have pores. Okay, they are porous. Okay, and then the next one is the presence of podocytes. Podocytes or filtration slits. Okay, filtration slits that allows substance such as water, urea, glucose, uric acid, ions to enter into the filtrate in the Bowman capsule. So those are the points that you can include in your answer. Okay, so for question number 7, okay, it says here, table number 1 shows concentration of solute in urine. Okay, in urine. So blood plasma and the glomerular filtrate. Okay, urine, blood plasma, and also glomerular filtrate. Three different fluid: urine, blood plasma, and filtrate. So the glomerular filtrate refers to the fluid filtered through the glomerular capillary into the glomerular capsule. So perhaps this is the Bowman capsule, okay, actually, of the renal tubule. So if you look at the table here, you have the solute that contains the filtrate or the fluid. Okay, the fluid that contains solutes such as glucose, protein, potassium ion, sodium ion, and urea for all the three fluid. Okay, urine, blood plasma, and also glomerular filtrate. So if you look at for urine, there will be no glucose, there will be no protein, as the protein and the glucose will be reabsorbed in the proximal tubule. Okay, will be reabsorbed in the proximal tubule. And then for the potassium ion, if you can see, okay, it is 1.5. There is potassium ion in the urine, sodium ion, and also urea. Okay, so for the blood plasma, there will be glucose, there will be protein obviously, kan? So the protein, the red blood cells will be maintained in the blood plasma in the glomerulus. And then you have potassium ion, sodium ion, and also urea. Filtrate, ada glucose, tak ada protein. Okay, filtrate, glomerular filtrate. And then there will be sodium ion, potassium ion, and also urea. Okay, so for question number 1, Siti Nur Shafika, can you read the question for number 1 and answer the question for that question number 1?
Explain the differences in the concentration of protein in blood plasma and glomerular filtrate.
Okay, the difference between the protein concentration, protein here, in blood plasma and also glomerular filtrate. Blood plasma, as you can see, the concentration is really high compared to the glomerular filtrate. So why is that? 2 marks.
Madam, for blood plasma, I don't have the answer, but for the glomerular filtrate, I answer because it cannot pass across during the ultrafiltration.
Why is that? Why does the protein cannot pass through the glomerulus into the Bowman capsule due to what?
The size of the molecule.
So the size of the protein or the molecule will be obviously will be. Saya nak keyword untuk dapatkan 2 markah tu. Sekarang ni baru 1. Dapat. Lagi 1 keyword yang saya nak. Protein size is what? The size of the protein, is it small or big?
Big.
Big lah. Okay, So if you look at the table, you can see that the blood contains a high number of protein. The glomerular filtrate will have no protein. Why? Because during ultrafiltration, the protein will maintain in the blood in the glomerulus due to its big size. Okay, big size. The big size of the protein will not enable the protein to get into the Bowman capsule to become part of the glomerular filtrate. Okay, so 2 point, protein in the blood plasma are present in the blood plasma but not in the glomerular filtrate. Okay, number 1, obvious difference. Second, because protein is not filtered. Okay, protein is not filtered. Number 3, because protein is a big molecule. Okay, due to its big size, it will not be filtered. It will not become part of the filtrate. Okay. So thank you, Shafika. So next for question number 2, Shirley.
For A2, explain how the glomerular filtrate is produced. My answer is blood flows through the glomerular capillaries under high pressure, forcing more than 10% of the plasma out of the capillaries and into Bowman's capsule.
Through the process, untuk itu baru 1 markah. High pressure tadi tu. Process what? The process of
ultrafiltration.
Okay, so dapatlah 2 markah. Okay. So I repeat again. For this question, explain how the glomerular filtrate is produced. So basically, you have glomerulus. Surrounding the glomerulus, you have the Bowman capsule. Glomerulus is blood capillaries. So the blood capillary of glomerulus will receive blood from the afferent arteriole and out into the efferent arteriole. Okay. So explain how the glomerular filtrate is produced through the process of ultrafiltration. 1 mark. Okay. Second, kenapa boleh jadinya ultrafiltration? Due to the presence of high blood pressure or high hydrostatic pressure in the glomerulus. Second point. Okay. Third point, kenapa boleh adanya difference adanya high pressure dalam glomerulus? Due to the diameter of the afferent arteriole is bigger compared to the efferent arteriole. Okay. So 3 point kat situ lah yang you can include in your answer. Boleh Can you all catch up?
Boleh.
Okay, so thank you. Okay, question number 3. Rashida.
Okay, figure 7 below shows a part of mammalian nephron. The number on the diagram show the changes in the relative concentration of solute in the filtrate as it passes through that part of the nephron. I explain how in the ascending limb, the relative concentration solute in the filtrate change from 1400 to 300. Okay, so firstly, the filtrate akan move to the uh, thick to the thin segment of ascending limb, and the salt and ACL passively diffuse out into the interstitial fluid. So this will help to maintain the osmolarity of the interstitial fluid in the medulla. And then the
The
filtrate
filtrate
can
can
move
move
to the
to the
thick
thick
segment
segment.
of the ascending limb, and the salt will diffuse actively from the filtrate into the interstitial fluid. The filtrate become more dilute.
Okay, so betul lah. Okay, so you got your point there. So betul lah. All you have all the points. So that contributes to 4 marks. Okay. In the question, so kita go through by semula. Explain how in the ascending limb of loop of Henle, the relative concentration of solute in the filtrate changes from 1400 to 3000. Dalam nota awak dia bagi 1200, lebih kurang lah kan. Now, the point is that the filtrate at the elbow of the loop of Henle, it is really concentrated because initially in the descending, there will be reabsorption of water. In the ascending, okay, in the ascending limb of loop of Henle, like what Rashida has mentioned, you have to significantly identify the two region of the ascending limb of loop of Henle. Kita ada thin segment, kita ada thick segment. Okay, the thin allows sodium chloride to move through passive transport. The thick segment allows the sodium to move out of the filtrate into the interstitial fluid through active transport. Sodium chloride yang keluar daripada filtrate, okay, pergerakan dia adalah actually daripada filtrate pergi interstitial fluid, and the sodium chloride actually will enter into the vasa recta. Okay, will enter into the blood in the vasa recta. Tapi dia akan keluar balik bila darah dalam vasa recta dia round into the ascending vasa recta sebelah dengan descending loop of Henle. So sodium chloride akan keluar balik semula sebenarnya. Okay, keluar balik semula daripada darah tu. Okay. But initially untuk soalan ni dia just nak awak explain how the ascending limb menyebabkan filtrate daripada concentrated jadi dilute disebabkan oleh pergerakan salt tu tadi. Passive and active. Passive in the thin segment, active in the thick segment. So that is the point that you have to include in your answer. Soalan awak ni, dalam awak punya soalan, 4 markah dalam saya punya answer scheme dia 2 markah. So tak apalah, kita ambil for the bigger marks. Okay. So another point that you could include in your answer is that if you mention in the ascending limb, the entire ascending limb of loop of Henle, there will be no reabsorption of water. It means that the water is maintained in the filtrate. Here, the water maintained in the filtrate, causing the filtrate to be diluted as well. Okay. So the ascending is impermeable to water. So that one you can also include in your answer. Okay. Okay, so I hope you got all the points. Saya note balik. Thin, passive untuk NaCl. Thick is active. Active transport for NaCl. The entire ascending limb of loop of Henle, there will be no reabsorption of water as it is impermeable to water. Another point that you could include in your answer. Okay. So question number 8, Izzatul.
For question number 8, the question is explain why glucose might be present in the urine of some people. My answer is glucose might not be reabsorbed in proximal convoluted said tubule. I'm, I'm not sure. Legend.
Kan kita belajar tadi ada berlaku reabsorption of after ultrafiltration kan, after ultrafiltration in the glomerulus to form the glomerular filtrate in the Bowman capsule, and then after that dia akan masuk region proximal tubule. In the proximal tubule, all these nutrients will be reabsorbed. Okay, will be reabsorbed. Sodium chloride, water, amino acids, protein. Not protein, but amino acids lah. Amino acid, glucose, vitamins, all will be reabsorbed. But okay, in the case where the urine of some people will still contain glucose is due to the intake of the glucose that is too high. Okay, the intake of the glucose that is too high and causing this excess glucose still present in the urine. Walaupun dah reabsorb dekat proximal tubule, but there will be also some glucose that can be present in the urine due to the high amount of glucose that are present in the blood initially. Okay. So the point that you could include in your answer, ini soalan ni berapa markah? Soalan ni 2 markah. Okay. So the first point, intake of food with high content of sugar or high content of carbohydrates. Second point, pancreas does not secrete enough insulin for diabetes patient lah kan, maybe. Okay, for diabetes patient, so the production of insulin may be low or there's no production of insulin. So there will be excess glucose in the blood that is not converted into glycogen and stored in the liver. Okay, so point yang ketiga, so therefore there will be high concentration of glucose in the blood. And then the next point, ultrafiltration in the glomerulus produces glomerular filtrate with high content of glucose. And next point, not all glucose will be reabsorbed in the proximal tubule. So it means that there is still some presence of glucose in the filtrate that becomes the urine later. So that's why for some people there is glucose present. Okay. So boleh faham Tadi siapa? Tadi Izatul lah.
Boleh.
Okay. So the next point, the next question, Fatiha.
Okay, B. Discuss why the blood glucose level of a person who is fasting does not show much decrease. My answer is body was not able to lower blood sugar level because the glucose is needed as a source of energy. Glucose in kidney will be reabsorbed back into the blood. Glucose will be transported actively from the filtrate into the interstitial fluid and then back to peritubular capillaries in the proximal convoluted tubule.
Okay. Awak tengok soalan ni Soalan ni dia adalah independent of question A. Okay. Question B is independent of question A. So it means that do not relate your answer with the formation of urine. Jangan. Okay, soalan ni apa? Discuss why the blood glucose level of a person who is fasting does not show much decrease. So it means that for this question, you must explain on how your body regulate blood glucose level through the involvement of the hormone such as insulin or glucagon. Okay, so itu je. Do not relate with the formation of urine because question B is independent of question A. Dia tak ada kaitan pun dengan urine formation. Okay, awak jangan confuse kat situ. Okay, question B adalah anak untuk question 8 saja. Dia tak ada kaitan dengan urine formation. So in this case, okay, the keyword in the question is fasting and causes not much decrease in blood glucose level. So when you are fasting, you are not eating. Okay, you don't eat rice. You don't take any sugar, for example. So your blood glucose level supposedly to fall below the set point. So if you refer to your notes, what is the normal set point? Ini awak kena refer pada blood glucose regulation lah in your note kan, which is in this slide. Okay, so the normal blood glucose level is maintained at 70 to 110 milligram of glucose. Okay, milligram of glucose per 100 ml of blood. Okay, so when you are fasting, you are lacking sugar. Okay, you are lacking sugar. So in order, sugar is needed for cellular respiration for that process to produce energy for you. Kan? So bila awak puasa, awak tak makan, you are lacking energy. So in order to get the glucose to do cellular respiration, to provide the energy for you, and to make sure that the blood glucose level increase back to the normal set point, so the hormone involved here is the glucagon. So awak kena cerita macam mana aras gula dalam darah initially dia fall due to fasting, increase back to the normal set point which is 70 to 110. Okay, 110 of glucose per 100 ml of blood. So mana dia nak increasekan balik? Through the secretion of the hormone glucagon. Okay, through the secretion of the hormone glucagon. Tengok ni, fasting, the glucagon will be secreted by the alpha cell. So what is the function of the glucagon? It goes to the target organ, which is the liver, and causes the liver to break down glycogen stored in the liver into its monomer, which is the glucose, and release into the blood, increasing blood glucose level to the normal set point. So itu awak kena faham soalan dia. Faham Jangan kaitkan soalan antara satu soalan dengan soalan yang lain. Kecuali kalau anak dia A1, A2, so itu akan soalan A2 akan berkait dengan soalan A1 lah. Tapi kalau soalan A dengan B, selalunya dia independent of each other. Selalunya. Okay, macam tu. Faham? Boleh tak?
Boleh je.
Okay. So yang ini fasting, decrease. So point yang awak nak adalah glucagon. Okay, glucagon, blood glucose level that increase back to the normal set point due to glycogen breakdown into glucose. So itulah point dia yang kita nak kat situ. Okay. Okay, so the next question, Fakihah, number 9. Ini kena draw. Okay, so question number 9, it says draw a level diagram of a nephron. So tak payah draw. Kat sini saya nak awak mention turutan structure nephron tu. Nanti baru saya akan draw. Untuk nephron, apa structure yang perlu ada dulu?
Glomerulus,
And then
proximal tubule,
sebelum proximal tubule.
Bowman's capsule,
Bowman capsule.
proximal tubule,
Okay.
descending loop of Henle,
Okay.
thin segment of ascending loop of limb of Henle,
Okay.
thick segment ascending loop of Henle,
Right.
distal tubule,
Right.
and collecting duct.
Okay. So if you have all the structures in the correct sequence, so I expect you to know the proper structure of a nephron. Okay. So you got all the 6 points here. So you just refer to the structure of nephrons. Ini tak berapa cantik sangat. Kita ambil gambar ni lah. Okay. So make sure. Susah ke nak draw ni? Will you find it hard to draw this nephron?
Yes.
Okay, so basically just draw it, just simple diagram. Okay, so here you have the blood capillaries of glomerulus. Okay, it receives blood from the afferent arteriole and blood masuk kan. So blood keluar melalui efferent arterioles. So here you have the glomerulus surrounded by what? Bowman capsule. Okay, Bowman capsule. Just ini bukan kelas art ya. Asalkan lukisan awak tu nice and neat lah. Ini saya tak berapa neat sangat. Okay. So this is the Bowman capsule. This is the glomerulus. Okay, and then you have the proximal tubule. Proximal tubule, maybe you can draw it a bit. Sebab dia convoluted kan, it is folded. So maybe, sebab nama pun convoluted, right? Okay, convoluted, folded, okay. So, and then into the descending limb of loop of Henle. So, and then the thin segment of loop of Henle, and then thick segment, okay, and then into the convoluted distal tubule, and then into the collecting ducts. Collecting ducts, okay. Kenapa ada cabang-cabang ni? Because actually it receives filtrate from several other nephrons. Okay, so your drawing must be properly labelled. Kena label baru dapat markah lah. Drawing and labeling contribute to the marks. Okay. Okay, so the distal tubule, collecting ducts, loop of Henle. Okay, loop of Henle. Alright. So the next question. Thank you, Fakiha. Next is Yasmin.
For B, name 3 solutes that are selectively reabsorbed back into the blood in the proximal convoluted tubule. Salt, glucose, and amino acid.
Okay, salt, glucose, amino acid, vitamins. Okay, salt ataupun cara lain nak cakap dia sodium chloride or sodium ion chloride ion pun boleh jugalah. Okay, so sama je. Okay, so dia nak 3, so you can mention any of the solutes. Okay, any 3 of the solutes. Okay. Next question, C. Siapa ya? Shakira.
Okay, for C, discuss why the blood glucose level of a person who is fasting does not show much decrease. This one, my answer is when fasting, the blood glucose level will fall, and then the alpha cells of the pancreas will secrete the hormone of glucagon into the blood. The hormone glucagon will promote the breakdown of glycogen in the liver and then release glucose into the blood. Thus, the blood glucose level will rise to the normal set point.
Okay, so macam tadi kan soalan ni exactly the same as the previous question stated here. So, the repetition lah sebenarnya, kan? So, so that is the answer lah, kan? So, blood glucose, when you are fasting, blood glucose level falls below normal set point. To increase back to the normal set point, glycogen will be secreted. Glucagon will be secreted by the alpha cell of islets of Langerhans. So it instructs liver cells to break down glycogen to glucose. Okay. So other ways of increasing apa lagi kesan glucagon ni selain dia break down glycogen to glucose? Apa lagi yang dia akan buat? Apa lagi kesan glucagon ni? Apart from it instruct liver cell to break down glycogen into glucose. Convert what? Convert
Amino acid.
Convert amino acid or fats into glucose. Okay, kan? So those are the point that you could include in your answer lah. If you refer to your notes, kan? Glucose tadi. Ini ini. Okay, slide ni. Okay, so you refer to this slide lah, kan? To know the function of glucagon, to know the function of insulin bila dia secrete, bila kedua-dua hormon ni dia secrete, apa target organ dia, apa target sel dia, apa kesan dia. Okay, so jangan lupa pasal amino acid and fats jugalah. Okay, so kalau insulin, when blood glucose level increase. So the this will inhibit the conversion of amino acid into glucose. Vice versa untuk glucagon. Kan? So glucagon convert amino acid or fats into glucose sebab nak naikkan balik glucose level to the normal set point. So you should know when these two hormone will be secreted and what is the effects of these two hormones. Ingat ya, hormon dia bukan enzim. Okay? Hormon adalah messenger molecule. Kita panggil dia sebagai messenger molecule that will instruct cell to do certain kind of reaction. Okay? Yang akan makinkan tindak balas dalam sel tu adalah enzim. Bukan glucagon yang break down the glycogen into glucose. It is the enzyme inside cell, but the glucagon yang instruct cell tu untuk buat those kind of reaction. Faham ke?
Faham, madam.
Okay, so nanti kita akan belajar detail lagi lah on hormone sebenarnya. Okay, kita on a different chapter. Okay, so D. Sufia.
What is meant by the term osmoregulation? My answer is a mechanism to maintain the osmotic pressure in the body by controlling water and salt concentration.
Maintaining gain and loss. Okay, gain and loss of water and control solute concentration. Itu poin yang kena ada lah. Gain water and loss to and also control solute concentration in body fluid. Okay. So that is osmoregulation. Sebenarnya kalau nak to be exact kan, it is the mechanism of organism regulate or control solute concentration and also balance of water gain and loss. Macam tu. Okay. So thank you tadi. Syakira ke tadi?
Sufia.
Okay, so thank you, Sufia. So the last one, Munira.
Explain why the blood glucose level fluctuates very little in a normal healthy individual. Because there's not much of falls and rises of blood glucose level, the blood glucose level maintained at 70 till 110 milligram per 100 milliliter. This lowers the breakdown of glycogen into glucose via glucagon hormone and lower the take-up of glucose into glycogen by insulin hormone.
Boleh lah, okay. Dapatlah markah. Okay. Soalan ni again 5 marks. There is a lot of marks there. Okay. So you have to detail out your answer. Okay. So the question it says here, explain why blood glucose level fluctuates. Okay. Fluctuates very little in a normal healthy individual. So it means that for a normal healthy person, the secretion of the hormones that regulate blood glucose level will be at the normal rate. So it means that there will be the two hormones, glucagon and also insulin, secreted by the pancreas to regulate blood glucose level. Point yang pertama, the two hormone. Okay, there will be hormone from the pancreas to control blood glucose level, which is insulin and also glucagon. And then the next point you could explain in your answer, you give the two different situations when the two hormone will be secreted. The first situation is when you said, well, when blood glucose level increase, okay, when blood glucose level increase, insulin secretion will increase. Okay, insulin secretion will be increased. Apa kesan dia? So this will what? Convert the excess glucose in the blood as into glycogen to be stored in the liver. One situation. The second situation is when you explain your answer, Untuk keadaan yang kedua, untuk secretion of glucagon. Glucagon will be secreted when blood glucose level fall below the normal set point to increase back to increase back your blood glucose level by breaking down glycogen into glucose. Faham tak? So you have to bagi kedua-dua keadaan yang mana kedua-dua hormon ni dihasilkan. Okay, so that's the end of discussion for this chapter. Okay, so